Supercooling advances human tissue preservation. The image from the confocal microscope shows that the sarcomeres, or muscle filaments, in engineered human heart tissue were preserved after supercooled preservation. UC Berkeley researchers have found a way to recover human heart tissue after it was frozen for 1 to 3 days. The researchers froze the heart tissue in a chamber that did not have any ice crystals. They did this so they would not hurt the cells. The researchers evaluated the structural stability of heart cells and assessed whether tissue functions such as autonomous beating and sensitivity to drugs and external electrical stimuli were preserved. A paper was published that said scientists were able to use a new technique on tissue that can help people in the future. The technique they employed, known as isochoric supercooling, was originally invented in the laboratory of Boris Rubinsky, UC Berkeley professor of the graduate school at the Department of Mechanical Engineering, bioengineering professor emeritus and a senior author of the study. To our knowledge, this is the first ever report of the supercooling and revival of an autonomously beating, engineered human cardiac muscle, said study co-lead author Matt Powell-Palm, a postdoctoral scholar in Rubinsky's lab. The results have near-term implications for the preservation and transport of organ-on-a-chip platforms. This will make it easier for more people to use them, not just those who can make them. Such platforms are valuable tools for the design of new drugs for diseases of the heart and other organs, he said. This study looks at what isochoric supercooling could do for people who need transplants. It also looks like it might be a way to save donor tissue and organs. There are 7 out of 10 thoracic donor organ that cannot be saved. This is because the organs cannot stay alive long enough to reach patients who need them. A donor heart is used to save people. A donor heart can be used for a limited number of people, but the amount of hours it can survive is not big. Currently, patients in Florida cannot receive a heart or lung from California because the organ would not survive the cross-country trip, said Powell-Palm. Addressing those challenges is one of the goals of a research center. Advanced Technologies for the Preservation of Biological Systems, ATP Bio, is funded by the National Science Foundation. One of the UC Berkeley professors in this research center is Kevin E. Healy, who is also a senior author on this study. Giving no space for ice. This is a hard sentence. It's tough for me to understand. But I know that it has something to do with freezing and staying in a liquid. The constant volume state that is characteristic of isochoric freezing requires the sample to be kept in a liquid in a sealed, air-free rigid container. This material doesn't allow ice to form at freezing temperatures, even if it is below zero. This is different from how people usually freeze things. They are in contact with the air at a constant pressure. Samples are frozen under atmospheric pressure. This is more energy than needed. In the past, they have been able to cool samples down to -22 degrees Celsius while keeping some of the material unfrozen. This is fundamental thermodynamics. When the material to be frozen is confined in a rigid box, then only part of the volume freezes, he said. Recently, Rubinsky and Powell-Palm discovered that isochoric freezing could be applied to the food industry. The isochoric process causes the food to stay fresh and good for a long time. Companies who use this process save a lot of money on energy bills. Isochoric supercooling is new, but it has been found by UC researchers, and Biopreservation Incorporated, a startup company, has been working on how to make this technology heartwarming result. In this study, the researchers used new cells from adults. These cells beat like a human heart. The system that they made was a small thing that has small tubes. It is like the way that people are exposed to food and drugs inside their body. It's not enough to say that these biological samples survived supercooling, said Healy. We wanted to demonstrate that physiological and metabolic function remained largely intact. The researchers put the heart on a chip in a cold room with an organ preservation solution. They took the heart cells out of it after 24, 48, or 72 hours and put them back in water at 37 degrees Celsius. Doctors looked at the heart tissue of people who had been cooled. They found that the cooling had not affected the structure of the heart. The doctors also found that, with longer cooling periods, there was a slight upward trend in heartbeat duration, but it is unclear how this would affect us. They found that beating of heart muscle in the human body chip resumed for 65 to 80% of chips that were cooled to -200 degrees. They also found that there was no difference between chips with different amounts of time in supercooled preservation. We consider these percentages relatively high for preservation recovery results, especially considering the intrinsic variability in these heart-on-a-chip platforms and the fact that no cryoprotectants were used, said Powell Palm. The researchers also found that after coming out of cold sleep, the heart tissue stayed sensitive to the medicine isoproterenol. The researchers said that they need to do more research to be able to make organs. The technology used to cool the tissue is sound and robust, but we now need to develop techniques to warm things up consistently. This was easier with the mini heart muscles that we are using for this study. Working on whole organs will require more work.